today we will be discussing about the isolation of DNA from plant material. So in the first video you have already seen that how DNA can be isolated from plant material. So today on the second part of the video we will be taking those isolated DNA and we will separate it in a agarose gel electrophoresis, and then we will visualize it by ethidium bromide staining. So on that note, so this will be done in particularly 3 phases, because DNA cannot be seen in the naked eye. For that you need to stain it. In this case we will stain the DNA with a fluorochrome fluorescent dye, ethidium bromide. You all know that ethidium bromide binds to DNA. It is a intercalating agent. It binds to DNA, and when we will give UV light, it will absorb at 300 nanometer and it will emit a light. Emission will be in the range of 590 nanometer. That means in the visible range, yellowish orange it will come. So by that way we can stain the DNA molecule, but now we cannot do this all of a sudden. So you have to run the DNA on a gel electrophoresis. In case of DNA we will be using agarose gel electrophoresis, and this is a horizontal gel electrophoresis. We will use agarose because agarose does not interact with DNA. So it is safe for separating DNA according to the molecular weight, because DNA is negatively charged in a agarose gel electrophoresis. DNA will be moving from negative to positive end in a submerged in a running buffer. So as DNA is negatively charged, it will move away from negative pole towards the positive pole. Now the migration depends on the molecular weight of the DNA. If it is a high molecular weight, it will migrate slow. If it is a low molecular weight, it will migrate fast. So like that way we will see the migration in a agarose gel electrophoresis. Now in order to load, so first step is the casting of this gel electrophoresis system. Second step is how we will load the DNA we have already isolated. So on that note we will be taking the DNA and we will adding them with loading dye. So loading buffer basically. So this loading buffer is composed of sucrose, bromophenol blue and xylene cyanol. So bromophenol blue and xylene cyanol, these are basically 2 tracking dyes. Bromophenol blue track around 300 base pair, migrates with 300 base pair of DNA, and xylene cyanol, it migrates with 4 kb of DNA. And we use basically sucrose so that the DNA does not float out, it co-sediments with the DNA, so that DNA moves along the bed of agarose. So in this video you will see number 1 is the casting of the gel, that is agarose gel electrophoresis. Second is the how we load the DNA in the agarose gel electrophoresis system. Then in the third phase we will run the DNA, and in the fourth phase we will take out those DNA and we will go and observe it in a chemidoc for the fluorescence intensity with the ethidium bromide staining. So I hope you will enjoy this whole process.
We will prepare 1% agarose gel, and by heating the agarose, we will prepare the gel apparatus by sealing both its side by tape. And place the comb at its position, and after preparing the gel we will let it cool for a minute and then add ETBR when the agarose gel reach a suitable temperature. After adding ETPR, we will slowly pour the agarose gel into the gel apparatus so that no bubbles form. Now we will let the gel cool down and solidify. This is the electrophoretic apparatus. We will plug in the electrophoretic apparatus. And then pour the running buffer into the apparatus. Now we will remove the comb from the solidified gel. And remove the tapes also, and then place it into the electrophoretic apparatus. You have to notice that the gel completely is submerged in the running buffer. Then we will mix the DNA into 5x loading die. After mixing properly, we will load the DNA into the wells of the gel. After loading is completed, we will start the electrophoretic apparatus at 90 volts for half an hour. After the run is complete, we will visualize the gel in a chemidoc instrument. And set the instrument settings for ethylium bromide. And let the image develop. So this is the developed image.